Hi, everyone. Welcome back to the Cerebellar Seminar Series. Today we have as our guest Devika Narain. Devika is an associate professor at the Erasmus University in Rotterdam, the Netherlands. She studies cerebellar and cortical circuits using environmental information to enable precise temporal control of movements. She was a graduate student with Eli Brenner and Jeroen Smeets at Amsterdam studying human motor learning. Then she was a postdoc with Mehrdad Jazayeri at MIT, where she became a neurophysiologist and she recorded from the prefrontal cortex, among other regions, and discovered the encoding of timing. Next, she returned to Amsterdam and was trained by Chris Dezeu and his lab in Rotterdam, where she became a cerebellar neurophysiologist, and that's been a tremendous benefit for all of us in the cerebellar community. Devika, it's great to have you today.
Well, thank you for that very kind introduction, Reza. And also thank you for the invitation. It's a really great opportunity to present our work to the community and get some feedback. Today I'm going to talk about some work that originally started out as a cortical, thalamic, basal ganglia project, and the cerebellum was nowhere in the picture. And I guess today I'm going to tell the story of how it came into the picture and what we found. So here was our original scheme. So there was the MFC that receives thalamic input, and the target of the particular area of the MFC that we were concerned with was projecting to the caudate. And what we were doing at the time was training monkeys to do context-dependent timing. So what that meant was the monkey would see either a red or a blue cue on the screen. After a random time, a flash would appear on the screen, which we would call set. And then if the cue was blue, the monkey would make a 1500 millisecond delayed saccade. And if the cue was red, the monkey would make an 800 millisecond delayed saccade. Now, if it weren't a circle, if the cue were instead of a circle, a square, instead of making a delayed saccade, the monkey would press a button, but the time intervals would remain the same. So there was this design, this 2x2 design, where the color indicated the duration of the interval and the shape indicated the effector. So it's a context-dependent timing task with two effectors. Now, monkeys were able to do this task. What you see on the x-axis is the production time of the monkey. What you see on the y-axis is the progression of trials. And what you see, I don't know if you guys can see my cursor. Can you see my cursor? Yeah, okay. On average, the monkey was producing 800 milliseconds and 1500 milliseconds was the target here. It's pretty close. Similar for monkey D. This is with a different effector. This is for the button press. This is for the delayed saccade. All right, what we do see, consistent with everything we know about timing, is that as the duration increases, the variability in the responses also increases. Weber fraction, scalar timing, whatever you want to call it. So what we're going to do is we're going to harness this variability. And what we're going to do is essentially bin these production times into smaller production bins. So all of the red lines that I'm going to show you now onwards belong to the short interval, and all of the blue lines will belong to the long interval. All right, so when we recorded from the medial frontal cortex of monkeys, so first, let me just break this down for you. So each of these panels is a single neuron. Here are 8 neurons. All I have done is I have taken the long condition and the short condition, and I've put the firing rate on top of each other. The y-axis, as you will find, is exactly the same. They're just one on top of each other. And the phenomena that I'm trying to demonstrate here is that if you take the endpoints of any of these red curves and you start to stretch them out, what you see is that they start resembling the blue curves. And this is a phenomena we call temporal scaling. We didn't, we weren't the first to discover it. Long and Fee, established this in the songbird. Joe Patton's lab has has found temporal scaling in the striatum. I do think we may be the first to show that this can be done flexibly. So on a trial-by-trial basis, the monkey changes from a short interval to a long interval in a context-dependent way, and you see that the neural patterns stretch and compress concomitantly. So that is the finding, and this is what we call temporal scaling, this stretching and compressing. So what we then wanted to do was quantify this at the level of the population. So we defined an index, we call it a scaling index, where 1 stands for perfect scaling and 0 stands for no scaling at all. And what you see over here is a 3D histogram where the peak of this distribution, the yellowest part of the distribution, lies between 0.5 and 1. So this is a population of over 500 neurons in the MFC. All right, so now we can look at the downstream targets in the caudate and establish the same pattern. So the peak of this distribution, the yellowest part, now still lies between 0.5 and 1. So that's not surprising, but if we now look at the antidromically identified input to the MFC, we see a very different pattern. What we see is that the peak of this distribution, this yellow part, now lies at the lower end. And in fact, that's also what we saw in the individual unit responses of the thalamus, that they weren't scaling, they were doing something very different. They were, in a graded manner, changing their amplitude. So to understand how this all could be, coming together and what could be the contribution of this thalamic input to the MFC to drive the temporal scaling in this flexible context-dependent manner, we turn to a framework that is now quite popular in motor control, the dynamical systems perspective. And here we take a vector X, which consists of the firing rates, the time-varying firing rates of n neurons in a population.
Devika, looks like
Yeah, sure.
Michael has a question. He's wondering if he could ask you.
Sure, Michael, go ahead. Go ahead. Please feel free to interrupt whenever you'd like.
Thanks. Hi, Devika. Can you hear me?
Hi, Michael. I can hear you.
Just to understand your previous slide.
Sure.
I have difficulties understanding your variance explained scale. I mean, typically we use it as a fraction between 0 and 1. So what is this number?
Yeah. So I didn't go into details, Michael. Actually, this is a random projection analysis, very much, very, very similar to dimensionality reduction approach, like a linear dimensionality reduction approach, like PCA. So the idea would be, in this case, what we do is we take the 500 neuron population, we would draw random vectors, project onto it, and compute scaling index for each of these. Then we would cluster these together, and that's what you see over here as a histogram. And variance explained is very simply, like the amount of, like you would have a principal component essentially, how you would basically calculate it against the entire population for these clusters. So it all adds up, all of these little cubes, these little clusters add up. So does that kind of, I didn't go into all of the details. It's usually I give a workshop about it, but does that kind of address what you're asking?
It does, yeah, thanks.
All right, sorry. Yeah, I know, just being very superficial here. Apologies for that.
I didn't quite understand it either. I wonder, what's the difference between, what's the message that we should learn from the right column that isn't there on the left? That's not in the thalamus, basically.
Right. So Reza, if you look here, so the idea is that then I was avoiding talking about variance explained because indeed I thought I wasn't going into those details, but this is the variance explained in the data of all of these little clusters that we are composing. We are breaking this population down into. These are the random projections that we are sampling across this population. And what you see is as the scaling index increases, the variance explained is also increasing. So, and that's what you see here in the caudate. So as so there's both a vertical component and a horizontal component, that as the scaling index increases, this distribution is also pointing upwards. On the other hand, the bulk of the peak. So there's two things. There's the peak of the population where it lies, and there's like what happens as this number becomes larger. And some of that you see over here, but it's a lot less. So that's the thing. It's not clean. Sometimes, when you're looking at the population, you will always find a continuum of a lot of different things. And this was our attempt to understand, where does the population as a whole statistically lie? And the argument here is that this distribution is lying on the lower side of temporal scaling. And it's broader, so it is explaining like its own chunk of variance explained in the data, and whereas this other distribution is of a different nature. Its peak lies on the different side of the spectrum, and as the scaling index increases, the variance explained is also going up. Does that clarify things a little bit, Reza?
Thank you so much.
Yeah, so the point is essentially that the thalamus is doing something different than the MFC. This is what we are trying to say, that it is unlikely that these inputs that are antidromically identified to the thalamus, it is unlikely that these are the ones carrying out the temporal scaling computation themselves. So this is the point. Thanks, Michael and Reza, for helping me clarify that. So now we're back into the dynamical systems perspective, where the rate of change of this vector, we assume, is given by these two terms, where the first term represents the local recurrent interactions within the MFC, and the second term represents external input. In our case, for the reasons I just stated in the previous slide, we believe are coming from the thalamus and are doing something different, we believe, computationally, than what we are finding in this very recurrent part of the brain. And I want to clarify, I am talking about the cerebral cortex, the medial frontal cortex. Okay. So I want to also clarify that we're talking about the system at steady state. These monkeys need 2 years to learn these behaviors. So the synapses, the weights are fixed. There's no active learning going on. It's a system where you give an input, it switches context, and it produces an output. So in order to understand how we could understand something like temporal scaling at the population level, let's do a thought experiment where we have these 3 axes, which represent the dynamics, the firing rates of 3 neurons. So the context is already on, the cue flash comes on, the monkey starts to time, the monkey makes a button press. So the monkey has made a response. Now, the argument is that if all 3 of these neurons are perfectly scaling, so if you take the red curve, you stretch it out, it fits on top of the blue curve, if that's happening in all 3 of these neurons, then you will find a camera angle. If I rotate this 3-dimensional frame, there will be an angle where I will have both of these trajectories line on top of each other. And I'm going to call that subspace. In this case, it will be a plane, but I'm going to call it a subspace, and generalizing to higher dimensional space, where these trajectories will align, and that means that they need to get to the end of that distance at different times. Therefore, it must be that they are moving at different speeds. So in this manner, speed becomes a code for decoding the ultimate time that the monkey produces. And so in order to identify this subspace in 500-dimensional space or n-dimensional space, we developed a dimensionality reduction technique, a linear one, analogous to principal component analysis, where in principal component analysis, the goal is to find the axes of largest variance. Those are your the eigenvectors that correspond to the largest eigenvalues. Whereas in scaling component analysis, you're looking for the axes of most alignment. So you're looking for the minimum distance. And there are some tricks to that, but essentially it's just about finding a different coordinate frame where you find this scaling subspace. So if we did this correctly, 2 things should happen from the data. All of the trajectories should line up across the different conditions. And second, from the speed in these trajectories in the scaling subspace, we should be able to decode the behavior, the time responses of the monkey. And we were able to do both of these things. So these trajectories are quite close to each other. And from all of these time bins, there is good agreement with the time the monkey produced. So now, well, long story short, there are a lot more things that we did in this particular work, which has been out for a while now. So we won't go into too much detail, but the idea is that the thalamic signal we hypothesized is organizing the trajectories along one dimension. So they're separating an amplitude. Whereas if I rotate this very subspace, what I see is this subspace that I was just talking about, that at the time we called the recurrent subspace, where everything is moving along the same path, but at different speeds. So they're just two sides of the same coin. And indeed, when we look at the actual dynamics of the MFC population and the thalamic population, you see that there's this alignment over here, and you see that this separation emerges in the thalamic population, admittedly more nonlinear than we had expected. So I'm going to kind of rename these and use these terms from now onwards. I'm going to call this alignment temporal scaling, because that is the basis of that computation. And I'm going to call this graded change in amplitude in the firing rate of units or at the level of the population. I'm going to call that amplitude scaling. Okay, so temporal scaling and amplitude scaling. And our hypothesis is that one is predominant in the MFC, even though we do see signs of the amplitude input coming into this population, but in the thalamic population, we expect to fully find more amplitude scaling. And so this is the scheme, and now we have attached some proposed computations to these areas. And now the question arises, why should the cerebellum be thrown into this mix? Well, I suppose it's this enduring suspicion that the area that we recorded from in the thalamus was in fact the ventral thalamus, and we cannot know for sure. It was stereotactically determined, and one never knows. So it was one of the motivations for me to become a cerebellar rodent physiologist, to be able to really be able to anatomically trace these circuits and test this hypothesis that could this context signal be arising in the cerebellum. And the prediction there would be we should find amplitude scaling in the cerebellum. Of course, we know that this is a loop with the MFC as well. So will we also find temporal scaling in the cerebellum? And that's the question that we're using, that we're kind of going forward with. And that brought us to trying to determine what is a good behavior where we could yoke the cerebellar circuit and the MFC related circuits, or MPFC in the mice. How could we couple these systems through a behavior where we had some confidence that these behaviors will give us readouts from these particular circuits? So that led us to explore the behavioral literature behind these areas, behind motor task, behind motor timing, and we discovered that we learned a lot, and I hope we have sufficient understanding, because we based our task on all of this understanding. But I see all of the experts in the audience here, so you guys can correct us if we got something wrong. But what we found was that there are fast and slow processes in cognition and in motor learning and adaptation. Sorry?
Here's a question for you. I wonder if you don't mind.
No, not at all, Reza, please. Did I already say something wrong?
No. Farzana has a question for you. I wonder.
Oh, Farzana. Okay. Yeah, sure, Farzana. Please go ahead.
Hi, Devika. Thank you. Yeah, so before we move forward, you talked about amplitude scaling, and I wanted to, in your set go task, what would be an example of that? Is it like you change the intensity of the go set stimulus? Like, can you explain a little bit like how we can get this amplitude scaling in your task?
So, well, if you're talking about the CHG task, the monkey task that I just showed you, the idea would be, so this is actually a real thalamic profile. I don't know if you can see it. It's just like a small thumbnail. The idea is that when we group the firing rate according to the production time of the monkey, and we look at the thalamic response, we see the same graded pattern. So the idea is that the firing rate of the thalamus sets the initial condition of the dynamics of the MFC.
Okay.
And so basically, when that initial condition is set, lower on the axis, for example, that trajectory will evolve faster along the scaling substrate.
Then I'm confused. Okay, now I understand. Sorry. So why would then we get higher amplitude responses in the thalamus for longer delays? That's not intuitive to me.
I see. So that is related to a separate model that we proposed, where we think that. So the idea is, I wasn't planning to talk about this today, but I'll go into it very quickly. If you look at the FI curve of a cortical neuron, what you would see is something like this, right? So that it saturates at some point. So if I increase the amplitude a lot, I reach the saturation point, right? So for unit change in input, I would get less than unit change in output. So the gain would be lower. In other words, the activity of the same neuron would unfold at a longer timescale. Whereas if the firing rate, let's say, of the context signal is low, so that would be the short interval, you may reach the dynamic range of the FI curve, and then unit change in input will lead to unit change in output. So that was the one mechanism that we proposed for why this could be plausible. It's still a hypothesis and remains to be tested, however. So
interesting, because we are recording PPC also with different intervals for visuals. And it's exactly that. When we increase the interval, the amplitude of the response goes up. So
really?
Yes.
Wow, that's very interesting. By the way, technically, you could also find that whole thing flipped, and that would be a sign that it's the inhibition mechanism that is at play, potentially.
Great.
You know, so that will give you a signal. Oh, that's very interesting. That's great to know. That work is published, so if you're interested, that we have the model and we can send it to you if it's useful.
Thanks.
Right. So where was I? Yeah, I was here, and I was saying that if you've read Daniel Kahneman's book, it's called Thinking Fast and Slow. And there he recapitulates all the research in cognition about how basically implicit learning is intuitive, automatic, rapid, whereas there is more effort that goes into explicit cognitive learning, and because it takes more resources and it takes more effort, it is a slower process. And of course, we know from Reza's work with Maurice Smith that when they proposed the very well established and famous dual rate model, that there's also a fast and a slow process in motor adaptation and motor learning. And subsequent work from Mazzoni and Kirkauer and Rich Ivory's lab, Jordan Taylor, Sam McDougall, and recently from Maurice Smith's lab, have all shown that the explicit learning, the explicit part of motor learning and adaptation is attributed and linked to the fast process, and it's the implicit learning that is considered to be the slower process. So and of course, we were approaching all of this very much from the perspective of understanding the neural substrates behind all of this. And we were also very glad to find clues in the literature to what this might entail. So the link between the cerebral cortex and explicit processes in motor learning and adaptation. And also there's a lot of discussion about how eyeblink conditioning and other implicit behaviors in motor learning are potentially cerebellar-dependent and are also expected to evolve on slower timescales. And so we study the temporal control of movement, especially in this case, we're interested in studying context-dependent temporal control of movement, which we believe straddles these two domains. And a priori, we did not know what this would follow. It's very different from the kind of visual motor adaptation that we read about in the literature. It's very different from the kind of cognitive tasks that we read about. At the same time, we wanted to anchor this to literature, and this is how we felt we should approach it. And this work is really led by two very resilient and talented students, Luca Mangili and Charlotte Wissing. And the way we wanted to approach this is we said, we're going to design the task for a mouse and make humans do it. So if you find some of the stimuli very strange, this is the reason. So we had fixed participants in front of some kind of a virtual reality setup, where we attached a custom-made AirPuff apparatus to the right eye of the participants, and what they saw in front of them was a tunnel. And so they saw a flash, which we call the Q flash, and after the Q flash, the tunnel started to move. We didn't want there to be any spatial cues. The tunnel was a very long tunnel. You wouldn't necessarily reach the end of it unless you waited for 10 seconds or something. And the Michelson contrast across the two contexts was matched. The statistics was IID for every time the tunnel was presented, and it was randomly sampled. So there was no spatial cue that could be used to learn or to hack your way into timing. You had to, we ensured that they used pure timing in these tasks. And they had to press, so the task, and the idea is that we would couple this task, which we think is an analog of the monkey task that I presented to you, which engages the frontal cortex and thalamus, with another task that we do in our lab, which we are pretty confident has cerebellar origins, called eyeblink conditioning. And the idea was to yoke these tasks together. So when the tunnel was dark, after the Q flash, the participant had to press a button around, or learn through feedback, learn to press the button at 800 milliseconds. And when the tunnel was light, the participant had to figure out that the desired interval was 1600 milliseconds. And if they responded within a Weber fraction controlled window, we deemed it a correct response. However, if their response lay outside this omission window, and here's the catch, they received a low latency periocular AirPuff to their eye. And this was the only feedback. This was the only error signal that the participants received. So just to remind you, in classical eyeblink conditioning, we have mice, and they're given these a quick flash of light, or any animal model, for that matter. There's a light in trace eyeblink conditioning. There's a transient flash of light, and after a fixed time interval, there is a unconditioned stimulus, or in this case, an AirPuff. And the idea would be that when learning, at the start, before learning occurs, the eye closes reflexively, whereas after learning occurs, even in the absence of the AirPuff, the eye closes, arguably to protect itself. The brain, develops a learning mechanism to close the eye in a well-timed manner to protect the eye. Now, in our task, there is no need to protect the eye. Inside the omission window, there is no AirPuff. So the omission window is to provide the participant feedback that now is the correct time. Anywhere, if you respond during this omission window, you've done a good job, and we take this as a correct response. So when we look at the eyelid traces during this task, as expected, on an incorrect trial, the AirPuff occurs. So the purple line indicates the time of the AirPuff. And basically the eye closes reflexively. It has a very stereotypical pattern. To our great surprise, on correct trials, this is the mean of the omission window. That 0 is the mean of the omission window, but the green color indicates that no AirPuff took place. So there was no AirPuff. If we look at the evolution from early in the task to later in the task, we see the evolution of what one might call a conditioned response. But this was very puzzling to us, because we had always heard from everyone in the eyeblink field that the eye closes to protect itself, that it's logical that one should, learn a conditioned response, a conditioned eyeblink. So this was very puzzling. And at first we thought maybe, they're pressing the button and the eye is conditioned to the button press somehow, and the eye is closing after the button press. And we found that there was no systematic relationship between the time of the CR onset, so it's always in the absence of the air puff, and the time of the button press. There was no systematic. Across two conditions, we had 30 participants and 27. So for 57 participants total, we found no real evidence of correlation between these two metrics. So just to look at the overall responses. So here on the x-axis, you see the trial numbers. It's the total trials that the participants did. 800, 1600 milliseconds were the desired intervals that they were supposed to learn. What you see is that their button press response converges. So it wasn't an entirely trivial task for them. It took them more than 200 trials to really get it. But you see that after that, the responses diverge quite rapidly. And only once these responses start entering the omission window were we able to, we only look at the eyeblink responses for the correct manual trials. So here we see the eyeblink responses, and what we see is that they more rapidly converge, to an asymptotic value, and they more rapidly converge to the desired time, which was again very surprising for us. So what we see here is not very surprising, because one would expect that over time the performance, the hit rate for the manual responses would increase. And of course, one would expect that after learning, there would be a well-separated, a distinction between the dark condition time and the light condition time. This is exactly as desired. What we really did not see coming is that what this means. This means essentially that on one trial, they are getting the dark tunnel, and the eye preemptively closes at 800 milliseconds because there's no air puff on a correct trial. And on the very next trial, when the tunnel is light, the eye preemptively closes at a later time, closer to 1600 milliseconds. So, and of course, the hit rate is also increasing, and there's no sign of extinction, because as you have more and more correct trials, you have fewer and fewer feedback training trials to help the eyeblink system. So all of this seemed quite surprising to us. Furthermore, when we looked at learning times, so when we looked at the time of acquisition of the eyeblink and the button press, we found that and we've tried a lot of different measures for this. We use the same criteria for the eyeblink and the button press. And what we find is that the time at which the eyeblink reaches a well-timed response for both conditions is earlier than that for the button press. And finally, we also made participants do a survey at the end of the main experiment, but before we did some control experiments where we studied voluntary blinking. And so after the main experiment, we asked them to report their awareness of different facets of the task. And what we found there was that their awareness of this conditioned response was lower than the reflexive response elicited by the air puff, and it was much lower than their awareness of pressing a button on every trial. So yeah, we tried to follow the methodology of Clark and Squire, and one does find that it's not like this is entirely zero. So there is some awareness, but there is diminished awareness. So it is relatively implicit. So I just want to clarify that we may be using the word implicit a bit too strongly. I won't talk about this today because of time, and so that we have plenty of time for discussion, but Luca and Charlotte also did a different condition, doing a strategy version of this task. So that is 30 plus the 27 in the strategy across 57 participants, we found conditioned eyelid responses that were context dependent and, changing on a trial by trial basis. The effective strategy was that it decreased learning time, but it did so for both the eyeblink system and the eyeblink responses and the manual learning task. But the CR time, the onset of the CR, was still learned faster. So that result remained unchanged, but both of them were accelerated due to strategy. And if there are any questions, please let me know. Just feel free to interrupt, and I'm sure I'll be able to stop and address them. Okay, so in part 2, I want to very quickly go over how we're planning to address what the neural mechanisms of this behavior could be. And I'll start with a very preliminary model and go into some preliminary data where we think we have uncovered some clues as to what might be going on in cerebellar cortical populations. So let's start with the behavior that we're trying to recapitulate. And what we see over here is essentially the CR time. This is a context-dependent change in the CR, and it needs to be sustained over the duration of learning. So how could this happen effectively? And this is just a hypothesis, just a computational model. We're still thinking about this, and we're happy to get any feedback if anyone thinks that there might be other plausible ways that this could be achieved. So what we have is a dark tunnel and a light tunnel, and we've done our best to take into account the visual neuroscience literature especially from the primary visual cortex to design these stimuli. In a way that it will mitigate some of the nonlinear effects that we see in primary visual populations. There is work from Michael Higley's lab that shows that L5 pyramidal cells in V1 are important for eyeblink conditioning. So if you take these out, then you impair eyeblink conditioning, and that these project to the pons. And we also know from this part, the primary visual cortex is sensitive to contrast and has nonlinear properties that allow neurons to encode contrast differently. So we expect that, if the signal arises there, by the time it arrives to the pons, there will already be some separation in the signal for the dark and the light tunnel. And as the signal that goes through the pons through the mossy fibers into the granule cell layer, we have several theoretical works recently that, and of course, all the way back to Mar Albus, where the function of the granule layer is believed to be that of a pattern separator. So using all of this, we quantify how much overlap is needed in order to achieve context-dependent learning on a Purkinje cell dendritic arbor. And so some of our modeling efforts have gone into this direction. So what we find is that we need a largely non-overlapping incoming signal from the granule cells that impinges upon the dendritic arbor of the Purkinje cell, and the idea is that the parallel fiber to Purkinje cell synapses for the dark conditions will be largely different than the parallel fiber to Purkinje cell synapses for the light condition. And after that, we know that the air puff in different tasks reliably induces climbing fiber elicited complex spike in the Purkinje cell. So the idea is that in this particular task, which we consider to be a bit the reverse of eyeblink conditioning, there will be a complex spike everywhere if there is sufficient error variation, trial and error. There will be a complex spike in various places except in the place of interest, except in the omission window, which will lead to, through the classical mechanism, depression of surrounding activity except within the omission window. Of course, the idea also is that these granule cell representations are carrying different amplitudes of signals. So there is a primary prediction that there's amplitude separation in Purkinje cells. There may be context-dependent amplitude firing rate separations in Purkinje cells already by the time this task is progressing. So that is a testable prediction. And of course, there are some predictions in the granular that we'll leave our granule cell colleagues to, if they're interested, test. And we previously proposed some mechanisms where there can be heterogeneity, different types of representations among Purkinje cells, and how these can be linearly combined to drive a coherent output to the cerebellar nuclei. And that's the same method that we would employ here to lead to a cerebellar nuclear output, eventually leading to an eyelid output that recapitulates the kind of observations that we find in the data. And essentially, as I said, today I'd like to focus on the prediction that cerebellar Purkinje cells or cerebral cortical activity in general would exhibit context-related signals in the form of amplitude scaling. So going back to the earlier part of the beginning of this talk about the conversation about the temporal scaling versus the amplitude scaling. And this project, which is still very much ongoing, and it's still very early, so I encourage you to take this with a grain of salt, because there is a lot that we need to still understand in these data. But it is led by Lucas Bayones, postdoc in the lab, and with a lot of support from Marit, Sasha, and Ache. And it is essentially the same task that we do in mice. Mice pull a lever at the right time, and they get an air puff that gives them this error feedback signal. That we have adjusted the times, the intervals for the mice. So now for the dark tunnel, it's 500 milliseconds versus 800 milliseconds. I'm happy to explain why these times are different for humans and mice. And essentially, what we can do here is that we can trace the area that we know from other work is implicated in in eyeblink conditioning. And we can trace that to the ventral part of the thalamus and trace that further into the cortex. And this is how we identified what the subset of MPFC that we were interested in recording from. And so the idea is that we have anatomically identified these different areas that we record from. So this is not really zero. So this is an electrophysiology session, or actually concatenation of some. But actually, these mice have been trained for months and months, for 2 months, I think, on average, 60 to 90 days. And at some point, when their behavior stabilizes, we start to do the craniotomy and start recording. And so here is an example session of a mouse that has kind of learned the behavior, and you see a separation of the timings, of the lever press timings. And when we record from the MPFC, we see many units. I'm just giving some examples here because we've not quantified this yet extensively, and we've not actually calculated the scaling indices and generated the kinds of histograms that I was showing you earlier. So there's still a lot to be done. But in general, we do find these patterns. The question is, what are the numbers and what are the exact characteristics of these populations? In the cerebellum, in the cerebellar cortex, and in many Purkinje cells, we see a clear amplitude separated signal. Again, there's a lot to do and there's a lot to quantify. They do tend to also show some scaling characteristics, so there is some mixed representation. We also find some signs of temporal scaling in the cerebellar cortex. And we can also do this, we can also quantify this at the population level. So this is, again, just a single mouse. First thing to notice is that the speed, so this is the amplitude scaling you see that the speed of the short interval is higher than that of the long interval, which is exactly what we have shown in earlier work. In the temporal scaling subspace, you see that the trajectories align well. In the context scaling, you see the amplitude scaling separation. So we find these patterns, but it's still very early, and there's a lot more. We find this in more than one mouse, but you know, there's a lot more that we need to investigate in this work. So still very preliminary, but enough for us to say that we make these observations, that we have observed both temporal scaling and amplitude scaling in cerebellar populations during this behavior. Yeah, and then with that, because I promised Reza it shouldn't be more than 40 minutes, so I apologize, I'm running a little bit late. But just to summarize, well-timed conditioned eyeblink responses change their timing. So they show remarkable sophistication. We hadn't expected eyeblinks to show this kind of cognitive flexibility, where the time of the eyeblink, conditioned eyeblink, can be earlier or later, depending on context. And what we also find is that we propose a computational mechanism for how context-dependent encoding of timing can occur in cerebellar Purkinje cells. And finally, we show very preliminary evidence for context-dependent signaling in single unit and population activities of cerebellar populations. But of course, this is ongoing work. And with that, I'd like to thank you know, my remarkable team, who are really the engines behind this work. And yeah, especially Luca Magili and Charlotte Wissing, who did the explicit implicit behavioral task. And Lucas Barones. Okay, I guess that's our son, who just came back from, I don't know if you can hear it, or Zoom is so I was just saying, thanks to the team for you know, doing all of this work, and for, you know, putting up with the roller coaster that science can be. And with that, I thank all of you for your time. I should probably mute my speaker.
Wonderful, wonderful. So it's we're open for questions, everyone. Please just go ahead and raise your hand, and I'll be happy to call on you. So I just brief question for you. It was really fascinating that the eyeblink response converged in humans faster than the manual response, indicating that, you know, this timing is being learned at potentially two different rates, possibly two different parts of the brain, maybe in the same, maybe in the cerebellum, maybe not. So did you see anything like that in the mice?
Yeah, Reza, we do find conditioned responses, but we haven't fully characterized that data yet. And we're still trying to understand it. It felt too soon to talk about it, but we hope that next time we'll be able to tell that story to you. It might, we're not sure yet. Let me say that way.
All right, very good. I think Rich, I think you had your hand up.
Yeah, I can't find the hand raising button, but thanks. So I just want to, that was quite a lot to think about there. They're puzzling, they're surprising results. So I just want to understand, they make these well-timed eye blinks on the omission trials when they're correct. But when they learn to associate the eye blink with the thing, that's all on error trials. And so on those trials, the timing of the air puff is highly variable. Is that correct? It occurs when they press the key, but on those trials, they're pressing the key quite far from the target because they're outside the window. Did I follow that correctly about how you build the association between the button press and the eye blink?
Sorry, Rich. There's been some noise here in my background, and I wasn't able to fully understand your question.
Okay, so the way I understand it, is they only get eye blinks initially. They only get air puffs when they're pressing out.
Just give me a second. Sorry about that, Rich. Sorry.
Okay, no problem. Childcare may call. Some preempts everything.
No, the signal to noise. It's been challenging. The background noise. So I really apologize, but could you just repeat that for me?
Sure. I just want to, the way I understand it is that the way they get air puffs is on the trials when they respond outside the temporal window.
That's correct. Yes.
And so those air puffs then are occurring with highly variable timing because sometimes it's too early, sometimes it's too late, but
Those air puffs are occurring. The time of the air puff is highly variable. That is correct. Yes.
but somehow out of that, that's how they build the association between the air puff and the key press, so that now on the correct trials, the air puff actually, I mean, the eye blink occurs pretty much exactly with the key press.
Yes.
And
Well, so I did show you this correlation between the key press time and the eye blink time, and there was no correlation there. So sometimes the eye blink, the conditioned eye blink, will come before the key press. Sometimes it will come after. That was our first suspicion, that could there be like some kind of conditioning on the button press, and it turned out to not be the case.
no, that's right. So, I mean, it occurs with the mean of the key press times, right? I mean, it was occurring around 800 and around 1600 milliseconds is when the blink was occurring.
Indeed. And there's sometimes a bias for some participants, but it's a systematic bias.
And I guess I find that I just wanted to hear your thoughts about how this very variable time when they're getting the air puff somehow then consolidates into a very consistent time when they don't get when it's a conditioned response.
So that is the model that we were, well, I don't know if you found it convincing, but the idea would be that above a certain baseline for the Purkinje cell, there would this air puff that would come at all of these times except during the emission window would result in some kind of depression, long-term depression of Purkinje cell activity around the emission window, almost like shaping it towards. And of course, we know from Mark Wagner's recent work and other work, although this is contested with some work from Kurt Holt's lab, if one believes in the existence of a temporal basis set, then one can imagine that, you know, there would be systematic silencing of large sets of input synapses that would potentially shape the response towards the emission window. And again, this is just a theoretical hypothesis, and you know, we would need some of the brave experimentalists to test it, but that is where we are at. That's how we're thinking about it at this point.
Okay. Well, that's very interesting. I'll let someone else go. Thank you.
Thank you very much. Thank you.
That was wonderful. And I think you answered Farzane's. Farzane also had a question about the protocol, and I believe you've answered it. If not, Farzane, go ahead and raise your hand, and I will come back to you. But did she answer it for you?
Well, I can wait until others ask their questions because most of my questions are clarification at this point, and I will come back. Yeah.
Okay, sounds good. Sevgi, go ahead. You had a question.
Yeah, hello. That was great talk. Thank you, Devika.
Thank you very much. Thanks.
I was wondering the details of the mouse task. Do they discriminate based on the cue, or are they different group of mice or different duration?
So yes, they do discriminate based on, so the same mouse gets both the context on a, well, not a trial by trial basis. We give them small blocks and we vary the length of the blocks. So yeah, but they are short blocks that they receive the context in different small blocks. But the same mouse does, if I understood your question correctly.
Yeah, I was gonna ask how they discriminate, like what type of cue are you introducing them for, short versus longer durations?
It's this dark tunnel versus the light tunnel.
I see. Same thing.
And yeah, exactly the same as the humans. We actually designed the task for the mice first and then made humans do it. And
Okay.
yeah, so.
Yeah, perfect. Thank you.
Sasha, you have a question.
Great. Yeah, that designing the task for mice and then making humans do it is spectacular. So the humans learn much faster than the monkeys. Takes monkeys 2 years to learn this?
Well, it was a different task. So the monkeys were associating colors with different time intervals. In that task, we had engaged the reinforcement learning system because we were giving juice reward, water reward. And so we very much wanted to avoid giving anything that engages the dopaminergic system in this case, so that we could have an error feedback system that is supervised and could engage cerebellar circuits. So this was the idea. So it's not directly comparable.
Okay, great.
The timescales, perhaps.
That's not actually my question. So my question is right at the end. You showed this lovely picture of what looked like potentially simplex and CRUS1 inputs to the dentate. Yeah. So my question is, do you know what, maybe it wasn't, but so do you know whether the dentate or the interpositus of mice is more responsible for this? I assume it's the dentate, and because like 500 milliseconds is a really long time for the interpositus, which is really much more concerned about getting the red nucleus to do the right thing than it is about the cortex. Yeah. So just wondering about that.
Yeah, Sasha, it's actually a very interesting area of it's the int DL. So there is a part of the in mice. Well, I guess, if you want, it's not really, it doesn't look very dentate-like. It is the lateral.
Yeah, I know what you're talking about, the DLH sort of thing, right?
Yeah, exactly. So there's a part that is just very dorsal, lateral to the main interpositus, but medial to the dentate. So there's a small, island over there, and that's really our target usually. And that's the part that that also you find is between VM and VL thalamus.
Right.
And I can't guarantee that, like, we don't record from areas that kind of go around and probably also project to some of these surrounding areas. But when we are doing our viral tracing and, our understanding of the kinds of electrophysiological locations that we're targeting, that is the projection to it. So it might be something in between.
Great, thank you.
But I'm not sure. So you think that the more medial, the more standard interposed would largely be driving the red nuclear response. In this particular case, I think there is a projection to the red nucleus, but we're talking about the thalamic pathway, the VMVL. There's a collateral that goes all the way up. It goes to M1, and the anterior collaterals go to M2, ACAD. So I think it's more a disynaptic pathway that we're concerned with, that is transthalamic, that goes and potentially sets the context in the MPFC. But yeah, it's an interesting point that you make about the timescales. And I know that there is, just before it reaches the thalamus, there is a projection to the red nucleus. So we've not thought much about that pathway. Do you hypothesize a role for that in this task?
Oh, I don't know. Well, maybe we could talk offline. Beautiful, beautiful talk, really thought-provoking. Thanks.
Thank you very much. Thank you.
Farzana, we can come back to you if you want to go ahead and ask your question.
Sure, thank you. So, Devika, I love the talk, but I should confess that I was very confused, and I'm
sure, happy to clarify.
Thank you. So I guess a lot of my confusion will be gone if you just go one more time through this concept of emission window and air puff. I think I just missed all of that part. Sorry about that.
No worries at all. I should have been more clear. So the idea is that normally in traditional eyeblink conditioning, the air puff will occur at the time that you want the eyelid to close, right? But in this case, the air puff occurs everywhere but that time. So we omit the air puff at that time, but it's not a time, it's a small window. And so that window in which the air puff is omitted is called the omission window. And the reason for that is, of course, in timing, we have Weber fraction related variability. So for longer durations, there is a larger variation in your response time. So to be fair, one would adjust the reward or punishment window. In this case, the anti-reward window, because you're being punished everywhere else. The reward is the absence of the punishment, the aversive stimulus, at least. So that's why it's omitted, and that's the omission window. Does that help clarify?
Well, yes, I had completely missed the part that the air puff is everywhere, and that's why you call that omission. And then just to make sure, so there is a dark tunnel and a light tunnel. The subject, whether mouse or human, is constantly receiving an air puff to their eyes.
Well, on every trial, if they respond at the wrong time, they receive an air puff to their eye. But then it's a very brief air puff. It's like a.
Then the air puff removed.
Sorry?
When is the air puff removed?
So it's a discrete thing. On a trial by trial basis, the air puff is administered for 60 milliseconds.
It's removed when the animal or the subject does the manual response. So if the manual response is
that's what you mean. Yeah, it's a transient air puff. So if you press the button and it's outside the omission window, you will receive a transient air puff for 60 milliseconds. Am I misunderstanding your question, Farzaneh?
I think I should just read your bioarchive paper's description of the method and not bother everyone here. But this
no worries. Just
the bioarchive paper.
Yeah, it is indeed on bioarchive.
Yeah, I will just take it. Thank you. Don't worry.
Oh, well, thank you so much for your question.
It's a brilliant design. It really is. So one more question. Go ahead.
I have a couple of questions more. So did you see any implication about the prediction during this mouse task? Do they predict the timing, and do they react faster and faster when they learn the task?
Well, yeah, in a sense, they learn through trial and error to respond at a reasonably well-timed, to give a reasonably well-timed response. What do you mean by prediction?
But like whenever they see the cue, do they produce earlier responses, or do they stick with the like 500 and 800?
Yeah, so within the mission window is symmetric around the desired time. So you will see that there is a fairly Gaussian. Maybe I should check with Luca and Charlotte, but I think it's pretty Gaussian distribution around the mean of the omission window. Yeah. So that, but if that's what you mean by preemptive.
Yeah. I mean, if they know it's a fixed time, they would be more.
Well, there's some variation for sure, and also more for the long interval because the omission window is also larger. There's scalar variability, and it's the same for the. We did find that the precision of the eye blink time was smaller than that of the button press, which was surprising to us.
I see. And is there any reason why they are on the wheel while pressing the lever? Could it be more difficult?
Well, I think we know from work in Javier Medina's lab, from Megan Carey, and also from Chris DeSeu's work, that mice just feel more comfortable in these kinds of, like in our standard eye blink tasks, they feel happier on a wheel. Megan has even showed that like if you motorize the wheel, they learn faster. We don't motorize the wheel, but we give them some comfort that between sessions or during breaks that they can walk. They have the freedom to walk around. There's no significance to it. We try to we have a hold on the wheel, and we even sometimes just ask them to, hey, like during the task, don't move too much. But it's just there for making them comfortable. Sorry, go ahead.
Together, like while they are pressing, releasing the lever, they were also walking. Could it be a little bit harder? I
think during training, no, I think so. We do try to make it comfortable for them, and I think during training they may be, especially in the beginning, allowed to walk when they are pressing. So they stop, press, pull the lever, continue walking. So we try to make it very natural for them. They get scared very easily. They get freaked out very easily. So we try our best to, in the beginning, make them feel comfortable. At some point when they, learn the task, we try to minimize all of these other interferences and all these other potentially confounding factors.
Okay, thank you very much.
Thank you.
One last question, Jorge. Well, we can't hear you.
Sorry, it was muted. Hello.
Hi,
Sorry.
hi.
Hi. Thank you for the presentation. It was really good. And I have a question related to the last part with the firing rate of the Purkinje cells compared to the other neurons of the cortex, and specifically with that of the spatial or amplitude or temporal modulation that you are refining. And I wonder if for the Purkinje cell case, it seems on the traces that you were having modulation of that firing rate in each exposure to any event, either the moment of the manual pause and as well of the moment where there's supposed to be the cue for the delay of the first airport, not airport, but the first stimuli, the second stimuli. And so have you seen if Purkinje cells then are essentially maybe responding to any kind of perturbation on the simple spike activity?
Yeah, so it is, as you say, very likely that there is transient modulation when the cue flash comes in. So one would fully expect that, suddenly the screen brightens up. It's a very salient signal. Probably some sensory transient will occur there. And certainly after the motor response is made, maybe even before, some kind of preparatory response. So you're right that the modulation is complex, it's multiplexed, and so some neurons will show just the sensory component, some neurons will just show some of the motor, some will show both. So yeah, all of that is true. So what we're finding more and more is like the kinds of general principles of mixed selectivity that were found in the cortex, maybe, and it's still too soon, we've not characterized it specifically, but at first glance, it would appear that some of these phenomena may also be active in cerebellar encoding of these kinds of tasks. So yeah, it's still a bit early to say. So I imagine you're suggesting some kind of mixed selectivity analysis or some quantification or some DPCA where you would do some epoch-aligned population analysis or some single cell. We always do GLMs, or we always do. I think our students have different preferences for different methods. But in this particular case, I think we've not really started doing the epoch-related modulation and trying to go into that yet. We've maybe a bit too singularly been focusing on the phenomena of temporal scaling versus amplitude scaling and quantifying that. But you're right, maybe we should do some functional classification as well.
Yeah, it's just that maybe in our hands and our experience as well, this kind of segregation into both amplitude or time on the level of Purkinje cells is confusing because maybe there's an in-between, which is they responding to any event. And then if they respond to any event, if the events are by themselves timely separated, then it will look like it is temporally modulated, or
but it is, yeah, but it would have to be the same idiosyncratic pattern that is stretched, right? So how would that appear as so there is a difference between something ramping at the same rate early and something ramping at the same rate late. And there's a difference between something ramping at a rate here and ramping at a very different rate here. So I think there would be some difference. There would be a way to distinguish what you say and
exactly
with real temporal scaling.
Yeah, that's my question. Exactly. So, okay.
Yeah. So there would computationally be a way to make that distinction, and so indeed.
Thank you.
All right. Thank you very much.
It's just wonderful. What a brilliant task design. And just as a just to thank you, but also suggest that maybe in humans it'd be really fun to try a reversal extinction, if you will, then spontaneous recovery, since you have two rates of learning.
Yeah.
You
Yeah,
think
yeah,
this-
okay, yeah. So exactly, like your paper with Maurice, the original one, yeah, there would be a very different prediction there. Yeah, and would we expect different rates of the slow process for the two intervals? Would you expect different rates for the slow process for the two intervals, or would you expect the rate of the slow process to be the same?
Yeah, I think that you would see spontaneous recovery faster for the first. To have spontaneous recovery would be very cool, but faster for the eye response than the arm.
Oh, you mean effector-wise? Oh, I'm sorry. I thought you meant interval. Yeah, for the effectors. Yeah. Oh, yeah. Yeah. That I agree. It would be
Yeah.
interesting if we found spontaneous recovery indeed, and in this case, of course, we would have to adapt both intervals. How would you do that, Reza? Actually, sorry to be
So I would change the
dense
cue
about it.
of light and dark
Yeah.
for the times and just reverse them,
Ah. Okay.
and then look at the look what happens after they've learned some amount of reversal.
But there's inherently different noise in the two processes. So one is a low noise process. So the shorter interval, the shorter duration will have more precision, and the longer interval will have more variability. Is that going to affect the spontaneous recovery?
I think if there is spontaneous recovery, then that suggests that each of those processes have underlying fast and slow. So the manual response
Wow.
and the eyeblink response are somehow independent mechanisms that they themselves have fast and slow. That would be one prediction.
Okay. No, that and also within, I was actually talking about the intervals. So when we switch the dark and light to light and dark, or let's say just we inherently switch the timing, one of these would start at a different, well, they would have different statistics at the beginning. So they don't have the same comparison point, is what I'm trying to say,
Yes,
to
I agree.
assess the spontaneous recovery. But I think it's very interesting also just to look at from the point of view of the effector, the eyelid versus no, thank you. That's a very exciting thought. And I think we should
You guys have done a really
reflect on
lovely,
it.
it's really beautiful design. Wow, thank you
very
so much. What a pleasure to start this cerebellar seminar with such an engaging talk.
Thank you, Reza. You're very kind. Thank you.
Have a wonderful night. Bye, everyone.
Thank you, everyone. Bye-bye.